your I released the exams. I think the class average was a 91, 92, 93. I don't remember. It was high, or it was good. So well done. I think the lowest grade was in the lowish to mid 80s, which is a good low grade to have, right? Like everybody did well. Have a look. I just opened it up where if you wish, you can go back through and for questions, or at least for the multiple choice questions, you can see what you missed. We will keep essentially that same format for the rest of the exams. I think that worked well. The exact number of multiple choice questions may vary some, but it will never be, just because I'd have to write this many, it'll never be more than like 40, I think. And short answer questions, I don't think it'll ever be more than, 8 or 10. But all the exams will be worth 50 points, just so every exam is weighted the same. In my opinion, the unit 1 exam is going to be easier. It will have been easier than maybe the next few you'll have. And I think most of that is because exam 1 took a broad look at some of the events. And then in units 2 and 3, we'll go into a little more detail about some of the mechanisms. For example, in cytokines, we went over different cytokine families. I think that chapter does well with clumping things together, paying attention to how things are organized, which terms belong in which basket. And again, if that's all you can remember, it may not, but that may give you a shot at reducing the number of options to guess from, right? So sometimes tests you can utilize deductive reasoning even if you don't know what the answer is right out. With what we'll do today, the complement system, it can be a little more challenging, right? So this is our first true blush at looking at a sequence of events that causes a response. In doing the complement system, there is an elegance to it, right? It moves from this point to this point. And what we've done, what I appreciated about this text, which is why I use it as the required one, is that they include a chapter on complement. A lot of survey style immunology classes don't. And I think that does a disservice because, one, I'm biased. Complement system is my jam, but also it's important. And there is a table in your text, and I think it's table 14.2, and I have it referenced in the slides, and it goes over but a small selection of diseases that are driven or that are a consequence of having a deficiency in complement components. We could spend an entire course talking about the consequences of inappropriate complement activation, and we allude to it in the chapter. And it's some of what I studied the bulk of what I studied as a grad student. But this idea that complement is so powerful that in things where you don't want complement activated, it gets activated anyway. And so ischemia reperfusion, so heart attacks, strokes, muscle injury, burns are good activators of complement. And then things like septic shock, which we introduced in the idea of these cytokine storms. But a consequence of those cytokine storm, a consequence of all of those bacteria, is that you'll also start to activate complement. And you're activating so much that the ways to control the amount that's produced or that is made active falls away. So that's why I like talking about it. But it's our first way to look at these sequential steps in these series of events, and that'll serve.
Recording in progress.
And that'll serve us well as we move into looking at T cell and B cell activation, where we'll have a series of events, and it just gets us thinking in that direction and the idea of a cascade. So let's do it. We should be able to finish always say this, we should be able to finish fairly quick, fairly early. And I think that's very useful, because what I would encourage you to do is start the process of kind of gathering and organizing your material. And we'll talk about the Unit 2 exam after we finish this. So what is the complement system? It is these series of interconnected proteins that and there are about 30. Many of them are soluble, but you can also find membrane-bound forms, especially when you're talking about receptors and some inhibitors. Complement system is named because what they found is that these things that were later identified as complement proteins complemented the activity of antibodies, and that was principally in the lysis of pathogens. And if you took what came to be known as complement away, antibody function wasn't as good, or wasn't as effective. Now, with that in mind, complement is, I would argue, is best known as an innate immunity mechanism. And in other classes, we might group this specifically with innate immunity. And really, that's a matter of convenience, because complement has an evolutionary history, right? We find complement components in different, and we find it, and it kind of has evolved as organisms have evolved. And so big player in innate immunity, and that kind of bears out in some of the ways that we activate complement, but it also plays a vital importance in adaptive immunity. And these are just the immune functions of complement. Complement seems to play diverse roles, both in immunity and in other things that are way beyond the scope of our class. And as such, it's a big place where a lot of interest in looking at affecting complement in some way may be a disease therapy. And so complement biologists are increasingly in high demand, because more and more clinicians and pharmaceutical type pursuits are starting to look to the complement system, because it has its hand in everything. Now, where you find most of the complement proteins produced are by the liver. And if you think back to chapter 2, when we talked about acute phase, and that these series of cytokines stimulate the liver to produce some of these inflammatory and innate protein, innate affecting proteins. One of them that gets produced is like MBL, and so that kind of ties the two together. Hepatocytes, can quickly produce many of these complement cells, but you also find complement proteins produced by other cells. There's brain neurons can produce their own to some degree, but generally we think of it being synthesized by the liver, and some inflammatory cells themselves can do it. So there are 3 activation pathways, and each activation pathway relies on a different serum protein binding to a target. And we'll think of complement, just for the purposes of being simple, as being something that gets stuck on the surface of something, or of a target or a threat or a pathogen. So we have 3 activation pathways that we already introduced when we looked at this in last unit. We have the classical pathway. And very simply, classical pathway is the C1, which stands for complement 1. The C1 complex binds to antigen antibody complexes. And so as you hopefully are drawing about, because we'll see it again in the next unit, remember, when you see an antibody response, it's best to think, oh, well, that's an adaptive immune response, right? Not exclusively, but for our purposes, mostly. The next pathway is the lectin pathway, and lectin is a protein that is capable of binding sugars to a variety of types. And so what MBL, which is the complement protein, does is it binds polysaccharide residues that are commonly associated with microbial surfaces. There is an additional activator. It's not just MBL. It can also be a protein called ficalin, and ficalin binds to a specific modification found on many microbes, and it's acetyl moieties, right? And so some texts, especially older texts, you'll see this called the ficalin, or the lectin ficalin pathway. I don't, but. And then the last activation pathway is one that's a little more nebulous. It's probably the one that was present first. So we're actually looking at them in order, in inverse order of evolution. Very likely classical pathway was the one that evolved last, lectin pathway before that, and alternative pathway likely first, and that is that you have spontaneous deposition. Complement gets gets spontaneously cleaved, and then this cleaved part sticks to a membrane. Sorry, I'm fighting off a tickle throat. Okay, so all of the pathways there's a sequential activation to these pathways, meaning protein 1 activates protein 2, which or protein 1 enables the activation of protein 2 and 3 and 4 and so on. And so what we get is the generation of these biologically active fragments. And what this does is it results in the accumulation of complement components that are being decorated upon the surface of the intended target, hopefully. Now, once you activate these pathways, regardless of whether it's classical lectin or alternative, what these all do is they converge at C3. And so often we call C3 the convergence point of complement because everything funnels to it. And then from the cleavage of C3, you get more of the downstream effects. Now, when you cleave C3, what you generate is a covalently bound, and generally, if you see the B after a complement component, what we are talking about is the larger part. There are exceptions, but generally B is the larger piece, and generally B is what's going to stick and stay and covalently bind to the target surface. So when you cleave C3, you're going to get two pieces, a very small soluble C3A. We've already introduced this in the last unit, and it is very inflammatory, and it is called an anaphytoxin. Think anaphylactic shock. And anaphytoxins are able to induce robust inflammation and immune cell activation. C3b, the larger fragment, is going to stick to the surface where it's being bound. And what C3b does is it is a part of the opsonization process, C3 and its other pieces that we'll look at in more detail. As an opsonin, we know that if C3b is decorated on a surface, a phagocyte will come by and say, "Oh, that looks like something I really want to eat," and it'll interact with C3b to induce phagocytosis. Now, C3 cleavage. So we're all the pathways funnel here, but what's interesting is one activation pathway can also amplify it, and that is the alternative pathway, because it relies on the presence of C3b stuck to a membrane. All of these pathways will induce deposition of C3b. So they all funnel to one place, and then one of these pathways can actually increase the amount that's being produced, which has important immune functions, but also could have pathological consequences as well. So once C3b is deposited on that surface, all of our activation pathways have converged, and from there we will eventually get the cleavage of C5, complement component C5. Again, C5 gets cleaved. Any activation pathway will lead to C5 cleavage, and you're starting to see a pattern. When C5 gets cleaved, you see the generation of a larger membrane or target surface deposited C5b. What C5b does is once it's generated, it fairly quickly recruits additional complement components, and you see them move in order because they are named based on the order of their discovery. So you'll see C6, 7, 8, and many, many copies of C9 will aggregate together, and they'll form this neat structure called the membrane attack complex, or the MAC. And formation of many of these MACs on a target surface will stimulate cell lysis of the target. All right, so that cell will die or it'll burst. And if that were, say, a bacterium threat, that particular threat's eliminated. When you cleave C5, in addition to C5b, you also generate C5a. C5a, for our purposes, acts very similarly to the way C3a acts, which means it's soluble. It's a robust inflammatory molecule, and it's another anaphytoxin. So for our purposes, we are going to focus on two anaphytoxins, C3a and C5a. So what are the biological functions? We've already introduced these, but you can really, for our purposes, we can neatly clump their immune functions into one of three places. They are going to, and overlap among them. It's going to cause target cell lysis, right? That's because the membrane attack complex forms and it leads to the formation of pores, which cause water to rush in, boom, cell die. It can cause inflammation because during the course of complement cleavage, we are generating these very inflammatory molecules that go and stimulate in addition to other parts of the complement system. So the anaphytoxins do it very well, but there are other complement components as they're being cleaved or broken down. They can stimulate inflammatory or immune processes. And then the last of our three characteristic functions is opsonization. And recall that is because we are going to have opsonins be deposited onto targets, which phagocytes will see, interact with, and induce chewing. The complement system then, at first blush, works best for the rapid response and elimination of threats of targets. So what we're going to do is we're going to take our pathways and just move through them, and we're going to look at the series of events that lead us to. So a little bit of background just on the classical pathway, and really this is just the background of the complement proteins. What's interesting about the classical pathway, it's the one that's probably the most recent addition to our complement immune system. But it was the first one discovered. And again, that was because people were studying these antibody things. They had this additional protein that, or additional series of proteins that seem to boost the activity of antibodies. Now, our complement proteins, although for the most part, they are named in the order that they are experienced in the pathways, they're really named based on when they were discovered. So there'll be a couple of places where they're out of order. But generally it follows in an intuitive path, which is nice. Now we've already introduced C3 cleavage and C5 cleavage, so we know B and A mean a breakdown product of some case. A and B are the most common, but we will also see other ones that will generate other pieces, and generally large complement fragments that get generated will be degraded further. Now, in our class, there is an extent to which we would cover this, but someone studying advanced immunology or actually who studies complement would get into many more degrees of degradation investigating those biological consequences. So with all that prefaced, the classical pathway has activators, right? And the major activator of the classical pathway are antibody antigen complexes, and principally this is, the antibodies that activate complement are IgM. And recall from chapter 5, what is IgM? It's this pentamer, has 5 antibody parts. It has a region that very well allows interaction with complement. And IgG, and certain subtypes of IgG work activate complement best. Mostly we are going to look in the context of antibody binding to something and being on a surface. It is also possible that because antigen antibody complexes can also be formed in solution, right? That's how precipitation and agglutination, or how precipitation works. It's interaction with soluble parts. Complement can be activated because antibodies that are forming these soluble complexes can also interact with complement. Normally these, excuse me. Normally these complexes are cleared by macrophages. And this is a symptom of some autoimmune diseases where you have excessive complexes can lead to pathology. The complement system tries to minimize that. Now, there are a couple of additional activators of the classical pathway. One is an acute phase protein we've already discussed called C-reactive protein, or CRP. CRP is adept at binding sugar moieties of many cell walls of many different types of bacteria and are principally a bacteria. And so C-reactive protein gets induced in that early inflammatory response as a means to try to quickly neutralize and eliminate threats. And one way it can do that is to activate complement. What's interesting is there are other activators. Some viruses have patterns on their surface that can directly activate complement in the absence of antibody. Interestingly, cells that are dying, necrotic cells and apoptotic cells, will express structures that the complement system can see directly. And it's thought that this process helps aid in phagocytosis, because we don't want these dying cells to exist for, especially a necrotic cell, we want it cleaned up as quickly as possible. Apoptotic cells, similarly, you have these cells dying. The complement system, by interacting with it, can tell phagocytes, hey, clean up over here. And then in certain disease settings, for example, amyloid beta plaques, which are a characteristic of protein aggregation in neurological disease, probably best associated with Alzheimer's disease. Amyloid beta plaques can directly interact with complement proteins via the classical pathway. But what we're going to use when we talk about activation is, you should know that there are other activators, but we're going to use the prototype, which is an antibody that has bound its antigen. Sorry about that. Why are you doing that? Can y'all still see my screen? Oh.
Yes.
You can still see it? All right. Sorry about that. The green box that shades it keeps going away. So if you stop seeing it, just send me a message in the chat or let me know or something, and I'll figure it out. Okay. So we're going to use antibody antigen complexes. So let's revisit, right? Which antibodies activate complement? IgM is arguably the best complement fixer, which means allows for activation, followed by IgG3 because it has the right region. IgG1 is maybe less so, and those are both much better than IgG2. So first blush, IgM and IgG. Second blush, which IgGs and in which order. Now, the other isotypes, the reason they don't activate complement via the classical pathway is they have no place for the C1, the protein complex that we're about to talk about. They have no place for it to bind, which means they can't activate it. And if you think about it, IgMs are that first antibody produced, right? And so they're going to be the first one produced. Say you're fighting off a bacterial infection and viruses, you have B cells that are being activated, you're going to produce some IgM quickly. Well, or quickly in the scheme of things, well, that IgM serves as a good place to activate complement to try to control the threat. IgGs are what are getting produced principally in the secondary response to bacteria and viruses. And for the most part, they are good activators of complement. Now, the C1 complex is actually a complex because there are multiple parts, all of which have C1 in it, and then different different alphabetical designations. The C1 complex, if fully formed, is C1q, and then 2 copies each of C1s and C1r. Right? So when an antibody binds to an antigen, C1q is the first complement protein that shows up, and it sees, once an antibody binds to a structure, it slightly changes the way that it's oriented. Its conformation changes, and what that does is it exposes these sites that C1q can now see. And there are requirements that we're not concerned with, but know that it takes sufficient antibody binding, these sites get exposed, C1q comes in and can interact. When C1q interacts with the FC region, 2 copies each of C1s and C1r are recruited to the area, and they bind, forming the complete C1 complex. And when this happens, the C1s proteins become enzymatically active. And more specifically, C1s is called C1 esterase. So it has, remember, ASE on the end is an enzyme. And so C1 esterase has enzymatic activity. C1 esterase, once it's active in the C1 complex, it's going to cleave C4. When you cleave C4, you're going to produce a soluble C4a fragment that gets released and has inflammatory functions, but we're not concerned with it. And then it'll generate a membrane-bound portion called C4b. So usually the thing that gets left behind on the membrane is the bigger part. So C4b gets deposited on the membrane. And when C4b is deposited, that now allows C2 to bind to C4b. So C2 is bound. C2 will also be cleaved by the C1s esterase. And so C1s cleavage of C2 generates, here we have our fragment letters flipped. We're going to produce a soluble C2b that's going to dissolve away and go do something. And then we're going to get a membrane-bound C2a. What this membrane-bound C2a does is it complexes, so it joins together with the other stuff that's on the membrane. And so what you get is the activation of the classical pathway leads to a complex on a target surface called C4b2a. And so if you were asked, all right, well, what complement proteins were cleaved to make it? We had the cleavage of C4 and we had the cleavage of C2. All of this cleavage is the result of the formation of C1, which includes C1q binding an antibody, and the recruitment of C1r and C1s. C1s does the cleaving. And so what C4b2a is called is it's called the classical pathway C3 convertase. A C3 convertase is something that will cleave complement C3. So what we've done is we've gone from binding of antibody to the cleavage of C3. And we look at how that works. Here we have C1q, which is it kind of looks like a flower or a tree, depending on how you do it. And it's bound to its IGs that have been bound. C1r and C1s are recruited. C1s becomes active and cleaves C4. C4, once cleaved, creates one fragment that sticks to the membrane. That allows the recruitment of C2, which C1s cleaves, and then you get the classical C3 convertase, which is called C4b2a. All right, everybody good? All right, so you just follow the order. One of the things that can help write is to just skip, just draw stick figures and arrows as you get used to the order of events. So the next activation pathway is the lectin pathway, and here we are looking at the major activators being polysaccharides, so sugar moieties on the surface of pathogens. Many of these are mannose residues, and so mannose residues will bind a complement protein called mannose binding lectin. So lectins are proteins that bind sugars, and this is a sugar that binds mannose pattern. We'll refer to this almost always as MBL. What are these residues? Where are these characteristic polysaccharide residues? You see them on many gram-positive bacteria, including Staph aureus. You see them on many gram-negative bacteria, including E. coli and Haemophilus influenzae, which causes disease in kids unless they're vaccinated. You see these mannose moieties on fungi, including Candida and Aspergillus, which are related to, which are pathological and cause infections, as well as yeast particles. And so, ficellin, the other protein that can induce lectin pathway activation, binds polysaccharides that have been modified slightly. And so the principal modification is acetylation. And so know that ficellin can activate. But what we'll do is we will use MBL as our principal stimulator. Because of what the lectin pathway is binding, it's seeing things, that are generally bad. So we know that those are PAMPs, right? These patterns that are maybe not terribly specific, but favor a foreign or non-self structure. And so the lectin pathway really is an innate pathway because it's binding in the absence of highly specific antibodies, which means the lectin pathway helps contribute to discriminating between self and non-self in the innate system. Now, the early steps of lectin pathway activation are MBL is going to bind to these sugar residues, and what's very, very convenient, MBL is analogous to C1q. They, in fact, look fairly similar if you see them when you see them on the next page. So the events of C1q kind of match up quite well to the events of MBL. Once MBL has bound to these residues, and there are requirements for binding, but let's say that they are all met, ficellin as well. Once MBL has bound, it allows the recruitment of two serine proteases, and these are called mannose-associated serine proteases 1 and 2, or MASP 1 and 2, which act in a manner very similar to C1s and C1r. And what we're seeing is that the classical and the lectin pathways are analogous to one another, but one favors innate detection via PAMPs, and the other favors adaptive detection via specific antibodies. And so once you have the recruitment of MASP 1 and 2, MASP 2 becomes active. It cleaves C4, generating the same fragments that the classical pathway did, which then allows the recruitment and ultimate activation of C2. These events are the same as in the classical pathway. It's just a different protease that's doing the cleaving. And what we get from the activation of the lectin pathway is the generation of the same C3 classical C3 convertase, which is C4b2a bound on the target membrane. And so we look, this is MBL, very similar, MASP 1 and 2, the proteases, they bind these sugar structures, recruit C4, which gets activated by MASP 2, it gets deposited, which recruits C2, which gets activated by MASP 2, until the point that we get C4b2a, which is our classical C3 convertase. Everybody good so far? Right, an elegance, I hope, or at least it can make sense. I like this picture from another text because it shows that the classical and the lectin pathways are, suspiciously analogous or homologous. And they're just coming at complement activation from two perspectives, principally an adaptive perspective and principally an innate perspective. And they can, and they themselves funnel into the same activation of C4 and C2, which leads to C3 activation. Now, the alternative pathway is going to be different, probably the one that was present first. I say that with some doubt, but not much. So the alternative pathway can be activated with any foreign surface that has the sufficient properties that allow complement to be activated. The alternative pathway is a key part of the early pathogen response in innate immunity. And why foreign surfaces do this well is that they allow the soluble activated fragments of complement to stick to them. And these include, what are some of these surfaces that allow these activated complement pieces to stick? Bacteria have receptive surfaces, especially those that are gram-negative that have endotoxins. They make nice places for C3 fragments to stick. Yeast cells, a neat thing that is used, I think clinically, but also is used for research, is called cobra venom factor, CSF. Some cobra venoms will induce complement activation, which contributes to the tissue damage at the site. Well, and yeah. Now, what's interesting is that some of the classical and lectin pathway structures, like apoptotic cells, viruses, aggregated antibodies, will also stimulate alternative pathways. So there is some overlap between them. So the early steps of alternative pathway activation, we have spontaneous fluid phase generation of C3b, which means complement C3 is being cleaved. And that's because complement C3 has this reactive thioester bond. And this occurs in the fluid phase because complement C3 will often exist as C3 water, and that contributes to it and all that. This fluid phase, and this is termed tick over because this is always happening. So there's always activation of C3. But here's and you can think of this as being the alternative pathway because C3 is always being activated in the fluid or in the fluid phase. Think of the alternative pathway as always being on, and what it relies on is just to stick to something that will allow it to stick, such as proteins and carbohydrates on cell surfaces. And you might ask yourself, well, that sounds bad. Like, our C3 is always being activated. Why do our cells not always get complement activation occurring? And that's because our cells expressed, mammalian host cells express many complement inhibitors that prevent unnecessary C3 binding or rapidly inactivate C3 that does bind to a place it's not supposed to. Microbes typically don't have these inhibitory proteins, or don't have the same numbers of them, but this is not always true. And I have a couple of slides later, but some microbes have evolved ways to mimic the activity of inhibitors to prevent complement action and elimination. Now, this soluble C3, what happens if it doesn't stick to something? Complement proteins are highly labile, which means they turn over very quickly. And so C3b that doesn't stick to something fairly quickly is inactivated. So we have this generation of C3b in the fluid phase. Well, once it sticks to a surface, and let's say it's sticking to a membrane that is receptive, when it sticks to that membrane, it allows the binding of factor B. So here we're starting to see, if you see factor B or factor D, your mind should immediately jump to, yeah, that's alternative pathway. Right? If you see C1, that is classical pathway, right? You can start to put these things in baskets based on where you see them. So we have C3b that's on a membrane that allows factor B to come in and stick. What happens is when this happens, this complex allows factor D, another alternative pathway portion, which has enzymatic activity. It actually cleaves factor B, which results in a soluble BA fragment, which dissolves away and has biological activity. But for our purposes, more importantly, it generates a membrane-bound BB fragment, right? Membrane-bound, typically bigger. And so what this does is that when you activate the alternative pathway, because some random activated C3b stuck to a surface, it can result in the formation of the alternative pathway C3 convertase, which is called C3b BB. And when you see BB, your mind should immediately jump, that's alternative pathway, so I need to trace back how could that have been activated. What's nice is that once you get to the formation of either of our C3 convertases, the series of steps moving forward will be the same, or will be similar. Now, a note about the alternative pathway, we've already inferred that it can amplify. The reason that the alternative pathway amplifies itself very well, but it can amplify activation through classical and lectin pathways is that, remember, all of those pathways generate a C3 convertase, which cleaves C3 into a C3b fragment, which means factor B binding can be possible via any pathway. And what that means is that the alternative pathway is good at taking one complement that's been activated and making it activate numerous times. And if you look from our simple schematic, we have C3 that's spontaneously activated in the fluid phase, generating the C3b, which is good at binding to things that will allow it to stick. And so in this case, it's binding to a pathogen surface. And this bound C3b makes is very capable of recruiting factor B. And then once this complex is formed, factor D is able to cleave factor B, which generates the alternative C3 convertase called C3b BB. Elegant, right? Now, when we look at the generation of C3, C3 cleavage is the outcome of all activation pathways. What this means is that the classical C3 convertase cleaves C3, resulting in C3A, which is our potent pro-inflammatory anaphytoxin. We referenced it in our overview and in chapters past. The deposition of a membrane-bound C3b. What that means is when we get that membrane-bound C3 as a result of the classical C3 convertase, that membrane-bound C3b actually forms together to form a C5 convertase. And a C5 convertase cleaves and activates C5. And if we were to look at the components that make that up, it is C4b2a3b. Right? We take our classical pathway C3 convertase, and we added the activated C3 piece, which is C3b. If you look at the, recall, how can we get to the classical C3 convertase? Either the classical or the lectin pathway. For the alternative C3 convertase, this cleaves C3 also, and it results in, again, a soluble C3A, which is our anaphytoxin, powerful inflammatory mediator. We get membrane-bound C3b, but in this case, we have a different combination because we have that alternative C3 convertase. And so what we get here is another C5 convertase, but it's comprised of slightly different things, and that is C3b, BB, 3b. Lots of these. And this C5 convertase does the same function. It cleaves C5, resulting in our downstream effects. And so just a note for organizing your mind, if you see a complex of complement proteins that end with 3b, it is safe to assume that it is a convertase that will cleave and activate C5. So a hypothetical is a famous scientist who happens to teach at Webster, teaching DNAP immunology, identifies a new protein complex related to complement. And I give you a bunch of numbers and letters, and it ends with 3b. The safe assumption is that it will be some form of a C5 convertase, because for our purposes, if it's a complex that ends in 3b, it cleaves C5. I was the famous scientist. And so looking at that convergence just in image form, whether it is the classical C3 convertase, how do I know it's classical? Because it has two proteins that are not related to the alternative pathway, C4b and 2a. It will recruit C3, which cleaves C3, giving us a C5 convertase, C4b2a3b, ends with 3b, so it's going to cleave C5, and that C5b fragment will initiate the terminal pathway, which we'll talk about in a couple of slides. And that's what's going to lead to more inflammation and ultimately cell lysis. In the alternative pathway, we have our alternative C3 convertase, C3b, BB. It allows cleavage of another not active C3, which results in the formation of a C5 convertase, right? Which C3b, BB, BB. But what it will also do is it will amplify. And we will talk about how that works, but you're going to have a protein molecule, a complement protein that comes in and stabilizes this alternative C3 convertase and allows it to cleave lots of C3b. So all of that C3 that's being cleaved, only one of them can form a C5 convertase. What happens to the rest is they themselves can deposit. And now you have, in this picture, you have 1, 2, 3 more sites where the alternative pathway can continue to activate. Each one of these C3bs could bind factor B and induce activation. So this is a very clear example for where you can get one activation can lead to the possibility for, multitudes more. And you can just imagine, well, what if, in this case, this C3 convertase, maybe it cleaves 2 C3s and one of those C3bs gets stuck. That place could activate and amplify via the alternative pathway. And so the alternative pathway in that way is good at amplifying all other ways, but especially the alternative pathway. And what contributes to this amplification through the alternative pathway is that the alternative convertase C3b-BB, it can and often will bind a protein called propyrdin. If you like keeping the complement designations, it's factor P. Sometimes you will just see it listed as just a capital P. And what propyrdin does is it's one of the only, and maybe the only one, positive regulator of complement. Complement is so powerful that almost everything tries to turn it down, except propyrdin. And when it binds to the C3 convertase, forming C3b, BBP, it creates a very stable, capable C3 convertase that can, spin through this cycle numerous times. So lots of C3 cleavage, which means you get lots of C3b, which means you have the opportunity for lots more alternative pathway C3 convertases, because all of these would be able to bring in a B and keep going. So when we get to the point where in either case, right, we're going to get C3 convertases, and their composition will depend on whether they are the classical lectin path or the alternative path, but both of them will cleave C5, which results in an anaphytoxin, which is this C5a potent inflammatory mediator, gets secreted, goes and does powerful things. And you also get membrane-bound C5b. C5b will lead to, when it binds its membrane, and it will bind in many places because these C5 convertases, I should say C5 convertase, sorry. Those C5 convertases are going to be great at cleaving through those C5s that are present. And so you'll get lots of deposition, or the opportunity for lots of deposition, which forms the membrane attack complex, or MAC. And what the membrane attack complex is C5b that is sequentially that has sequentially added to it C6, C7, C8, and then C9. And C9 is important because it is a polymerizing subunit. Many copies of C9 are going to come flying in. And you can write the membrane attack complex as C5b 6, 7, 8, 9. Sometimes you'll just see it C5b-9. And what this complete membrane attack complex does is it forms a pore. And you're going to get these collection of pores on a target surface from all of this complement activation. That's going to allow water to rush in, ions to rush out. Osmotic balance is bad. The cell lyses, the target lyses. And here's what that looks like. C5b, in this case, it's already bound to the membrane. Sometimes this aggregation occurs just floating right off the surface. But regardless of how it happens, C5b 6, 7, 8 allows multiple copies of 9, and it forms this nice channel by which water floods in and ions flood out. I added a couple of papers. Paul Morgan is a guy that we tangentially collaborated with, or at least I did. And these are very cool methods and these papers are fairly old, that are looking at the membrane attack complex. And this one's nice because what it's doing is it's rotating you through what that membrane attack complex looks like. You can see kind of the half ring. So these would be phospholipids that would be continuing. You have a pore. I think you think back to the channels that we talked about in cell biology. In this case, these are unregulated channels that are just allowing water to move at will. But then if you look at panel A and B, these are nice illustrations. This is panel A. These are all membrane attack complex sites. These are all MAC sites on a target surface. And look at all the number of those pores, right? That's going to cause lysis fairly quickly. And if you get closer with more magnified microscopy, you can see the C9 structures very clearly. And so powerful forming these pores, which are cool to visualize. And this is actually fairly challenging because it's hard to visualize membrane-associated proteins. So I borrowed this from another text, which is a nice, and there's a little more detail, right? We're looking at soluble water. You can simplify it to fit based on what we've talked about. But the moral of the story is classical pathway activates through bound antibody and others, eventually leading to the formation of a C3 convertase, which cleaves C3, which induces effects, one of which is to activate C5. Lectin pathway is the result of PAMP recognition, which are, in this case, are going to be polysaccharide residues, will be activated by MBL and or ficillin, and then follows a series of events that are homologous to what you see with classical pathway, leading to the same events. And then the alternative pathway is spontaneous hydrolysis, where complement gets activated in the water. That activated C3b fragment can stick to a favorable surface, and then it results in alternative pathway consequences. And one of those is to amplify complement activation. Now, all of that, and that leads to our effects, which we'll talk about after we talk about regulation. With the powerful nature of complement activity, lots of investment in regulation is done. If you poorly regulate complement activity, not good. You will get things like inappropriate inflammation and lysis, which means normal tissues are going to be destroyed. Excuse me. More important, more concerningly in the short term, I guess, is that if you're not regulating complement activity, you're allowing too much of your complement to be activated. And what that does is it reduces your immunity, because now you don't have enough complement to proteins to be activated in the correct place. And so it increases susceptibility, and you'll see that merited out when we look at complement dysregulation and deficiencies. So dysregulation can also contribute to other things beyond susceptibility to infection. Inappropriate activation is a result of, or it's a consequence and drives some tissue injury following ischemia reperfusion, so heart attacks and stroke and many others. It plays a role in autoimmunity, especially maybe the characteristic is rheumatoid arthritis and among many, many others. And then damage to the retina is actually a consequence of dysregulation in age-related macular degeneration, or wet AMD. Now, in a healthy immune system, this dysregulation is fairly rare. One, that's because we express inhibitors, we express inhibitors, which minimize inappropriate complement activation. And that's important because that helps our host cells not be targeted, because complement can be indiscriminate in the absence of an inhibitor. Microbes, for the most part, do not share that privilege, and so they are going to be more susceptible. Now, these inhibitors are can be either membrane bound on the host membrane, and then you have some that circulate and are soluble, right? So we're going to get inhibition from two different perspectives. And we're taking a fairly argue superficial pass, but I think it's important to identify the places. And so what I tried to do was organize it in a way that made sense. What are some of the inhibitors of the classical / lectin pathway? You have those that can affect activation, or I guess inhibit activation. Maybe the best characterized of these are the C1 esterase inhibitors, or C1IH. It's a serum protease. And what it does is it actually breaks apart the C1 complex. And this is this is a complete C1 complex. And this is, in the presence of the inhibitor, what it does is it pops out C1s and C1r from C1q. And when this occurs, it makes inactive the C1s part. C1 inhibitor, because of the homology between lectin and classical pathways, C1 inhibitor does the same thing to MASP1 and MASP2 from MBL. Now, your text references the fact that there's likely a role, or maybe a designated role, in the alternative pathway, but we're going to ignore it. Smiley face. So C1 inhibitor interferes with lectin and classical pathways. Now, another thing you could do in these pathways is to decay or help to degrade the classical C3 convertase. And a reminder that C4b2A. There are 4 proteins that we'll concern ourselves with that do this. C4 binding protein. How should you know that that's related to classical and lectin pathways? Because both the classical and lectin pathways have C4 as a thing that as a protein in the cascade. Another one called decay accelerating factor, or DAF, complement receptor 1, also known as CR1, and then membrane cofactor protein, or MCP. All 4 of these are widely expressed on host membranes, and what they do is they interfere with the binding of C4b to C2 and C2a. What that does is it either prevents formation of that C3 convertase, or if it's formed, it helps to cause it to fall apart. Now, one of the ways it does that, or how some of these do that, is through C4b inactivation. And this is achieved by a serum protease called factor I. What factor I does is that once you have popped off C2a from the C3 convertase, this now free C4b is an attractive binding partner for factor I, but it only will do that in the presence of some of these other inhibitors, and they are collectively called cofactors. And these include C4b, MCP, and CR1. And what factor I does in the presence of these cofactors is it cleaves C4b into further degradation fragments that do not allow complement activation to continue. C4c and D, you see the pattern. Just a note to say, DAF is not a cofactor for factor I. What DAF does is it just interferes with the interferes with the association, but it doesn't induce cleavage through factor I. And so we have an image where we see some of these span out. There are different structural relationships between these all, but for our purposes, I'd rather you know what they do than the way they look and the specific mechanism. And when we turn to our alternative pathway, we can look at the inhibitors of this. Another thing we could do is quite simply decay the alternative pathway. All right, the alternative pathway C3 convertase. And this is achieved by another serum protease called factor H, or a serum protein called factor H. And what factor H does is it actually is a competitor to factor B. So when you have the C3b bind on a host surface, factor H and factor B are competing for this binding. And should factor H be the one that binds, it will, one, prevent factor B from sticking, or if it's already stuck, it's such a better binding partner that it can kick the BB fragment out. So why is it then that host cells are not attractive places for C3b to stick? Well, that's because host cell membranes are high in sialic acid, and sialic acid is a good recruiter and allows lots of articulation with factor H, which means around host membranes, there is just much more factor H present, which means when C3b sticks, factor H is what's going to bind quickly. Now, if you have a C3 convertase, alternative C3 convertase that's already formed, factor H will come in and is capable of kicking the BB fragment out. And when doing so, it's going to behave like a cofactor. And so when it does that, factor H will allow factor I to come in and cleave. So we have multiple cofactors for factor I, some from one from the alternative pathway and some that are more related to the classical pathway. What's interesting is that when you cleave C3b, so when you inactivate bound C3b by factor I, you get some of these degradation fragments, which will play a role in some of our biological functions we'll continue with. And the order of events is it gets cleaved to IC3b, then C3d, and then eventually C3dG. And collectively, these three things are all considered opsonins, are part of the opsonin profile. And these are not only opsonins for phagocytosis, but they are also attractive ligands for some of our complement receptors. Now, based on the function, MCP and CR1 are good cofactors for factor I, but they don't necessarily discriminate. So you can see MCP and CR1 will also allow factor I to inactivate C3b of the alternative pathway. And just another reminder, DAF does not stimulate factor I activity, but it can also interfere and cause alternative C3 convertase to fall apart. And we can see that illustrated here. Now, how do we then prevent lysis from forming? How do we inhibit the terminal pathway, which is what results in C5 cleavage and maybe that. So we could block C9 polymerization. All right, we could keep multiple copies of C9 from forming. The best of these are CD59. CD59 is widely distributed on host membranes, and what it does is it prevents additional copies of C9 from polymerizing. It can also prevent any soluble membrane attack complex from sticking. So if you have C5b through 8, which hasn't stuck to a surface yet, it formed in solution, it will have a hard time. And I like this other text uses an umbrella to show like it keeps it from being able to stick. You can also, you also have some soluble proteins that are good at keeping the MAC from sticking to a membrane. And this would be the case that if you have membrane attack complex proteins, or C5b, or any of the combination of those that have not yet stuck to a membrane. These soluble proteins, clusterin and vitronectin, are able to clump to those to keep them from sticking. And what that does is it spares lysis. So just to repeat, our biological activities are opsonization, inflammation, and cell lysis. Opsonization and inflammation in large part are driven by other cells being able to see that complement has been activated. That's going to be through complement receptors. So in opsonization, receptors on phagocytes will bind these opsonins, and they are the deposition fragments C3b and C4b, including some of their inactivation fragments, which include C3d, DG, IC3b, and the I here stands for inactivated. IC3b and another, and I included this here, C4d. We care very little about C4d. What about inflammation, right? Those anaphytoxins get produced. How is it that they are inflammatory? Well, they need to be seen by specific receptors on inflammatory cells and other cells. And the receptors for this are very simply C3A receptor, C3AR, which only binds C3A, and C5A receptor, which binds C5A. Your receptors are very neatly summarized. And again, my picture, it was too big a table and the lighting wasn't right when I was putting it together. But again, refer to table 14.1 to get a nice, clean listing of the receptor, what its function is, who it binds. Right? So when looking at the biological activities, they can really be broken up: opsonization, anaphytoxin production, and cell lysis. So opsonins, you could argue that opsonization is maybe the most important. I would argue that and inflammation are the most important. Complement activation, so the production of opsonins is a sign that complement has been activated on this target, and that is not good, or that this thing that has this evidence that complement has been activated is bad. And so opsonins include C3b and C4b, right? Those are features that complement has been activated, but it also includes some of these downstream degradation fragments. And I'm going to use C3b, which gets inactivated by either factor I or factor I gets cleaved into IC3b, C3d, and then eventually C3dg. Why have these degradation fragments? What this allows is that you can stop complement activation, right? By degrading C3b, you no longer activate complement, but you don't want the immune response to stop. You want complement activation to stop because it's powerful, but you want there to still be a tag that says, hey, come get me. And so by having these degradation fragments that are opsonins, it facilitates removal, right? Come get this carcass. We have marked it as dangerous. Come clean it up. And so what this really does is it helps control complement activity. It keeps complement from being too active without sacrificing necessarily an immune defense, right? Which I think is very elegant in that way. So what do opsonins bind when they're deposited? They bind complement receptors largely on phagocytes, inducing phagocytosis. Macrophages and neutrophils, while any of our phagocytes will express them, and in other contexts, we would talk about dendritic cells, but we'll focus on what's cleanly listed. Macrophages and neutrophils express lots of three particular complement receptors: CR1, CR3, and CR4. You have seen CR1 before. Recall, it helps, it's an inhibitor of complement activation, but it is also a receptor, at an opsonization type receptor. Tissue resident macrophages, so in different places, they might express slightly different things. Here, they'll express complement receptor IG, which is a immunoglobulin superfamily complement receptor. And we are not getting into the weeds necessarily. Your table does a good job of illustrating which receptors bind what, but each receptor has small differences in the exact opsonins that they are capable of binding. What that does is it gives you lots of redundancy so that clearance can happen. It also allows for different cell populations to respond differently. Now, in our biological, or in our next biological activity are the productions of anaphytoxins. Again, these are those soluble cleavage fragments, C3A and C5A. Those are the ones that we are focusing on. And what they do is they stimulate cells through C3A receptor and C5A receptor. What's interesting and not covered in our text, but just so you know, C3A and C5A are very rapidly degraded. They are rendered inactive. Now that has consequences, and those fragments can be done. That's beyond the scope of what we want to talk about. But these enzymes rapidly inactivate, and that helps control the inflammatory profile. It also encodes for other responses. Now, these amphitoxins are potent inducers of inflammatory and innate immune responses and other things. All right. So we're taking a very simple view of it. Now, what do C3A and C5A do? They act on vascular endothelial cells. That this action causes an increase in permeability and allows fluid to leave the blood and accumulate in the tissue. Right, that part of that inflammatory response. C3A and C5A are good chemoattractants, especially for neutrophils and other phagocytic cells. So you have more of these being produced in an inflammatory response. Will come over, follow us, and get ready to eat. They also induce smooth muscle cell contraction, which aids in the accumulation of fluid in that location. And on basophils and mast cells and other granulocytes, they also stimulate degranulation. So C3A and C5A, as a part of that anaphylactic shock, they are good at stimulating basophils and mast cells to release histamine. So histamine is, again, one of those key proteins that drives the inflammatory response in our 4 cardinal signs. And we can see that just kind of listed here or illustrated here. And just a brief word, we've already talked about a membrane attack complex that results in cell lysis, forms these pores, creating osmotic imbalance. You get lysis of the target. Now, there are some other biological activities of note. Some of these we will talk about in the context of different responses. One pretty important response is that complement plays a role in the B cell response. Complement activation is an enhancing signal and in B cell activation. And if you think about it, this makes sense, right? This bacterium must be bad. Why must it be bad? Because it is interacting directly with a B cell receptor, but also it has complement deposition fragments. So it's been proofread, right? If the complement system saw it as bad, and then we as a B cell see it as bad, it must be bad. And so especially C3D and DG fragments, when those are decorated on a surface, they will actually engage complement receptor 2, which is expressed as a part, also called CD21. And for complement stuff, we'll focus on CR2. When we talk about B cell receptors, we will probably switch to CD21, so be able to move between them. But CR2 in this way is part of the co-stimulatory process. Complement fragments are a sign that says, yes, this thing is bad. And BCR binding and a complement tag, lots of activation. And the way this works is that, the general scheme behind it is a B cell that is seeing an antigen that has this complement activation tag in the form of C3D or C3DG is a sign to say, yes, do it. And you actually see that the threshold, the activation response is 1,000 times greater if you compare it to a B cell activating antigen without complement deposition. Now, another B cell contributing to these B cell responses, CR2 binding also enhances B cells and their antigen processing, which improves their response. And it seems that memory B cell development is benefited by complement activation. Follicular dendritic cells that are in germinal centers, and a reminder that germinal centers are in secondary lymphoid tissue, and they are sites where B cells are undergoing their affinity maturation. They're getting better at making good, strong antibodies, strong binding antibodies. And this is mediated in part by CR1, CR2 to allow some of these memory cells, memory B cells to form. The mechanisms are beyond our scope for now. Another thing that complement contributes to is the clearance of immune complexes. Antibody antigen complexes, yes, often stick on surfaces, but they can form, especially when too many of them are forming. They'll start to clump together in solution, so they'll precipitate. If these precipitates get too large before they're cleared, they make for a surface where they become inflammatory and detrimental. Again, a symptom of, for example, lupus. You have lots of these autoimmune antibodies being produced, and they allow clumping, and then these clumps promote damage, especially in the kidneys, where you get immune activation. So what the complement system can do is, when these large antigen antibody aggregates form, complement can be activated, especially alternative pathway. C3b can be deposited, and what it can do is, one, it can interfere with the clumping. So it interferes with the bonds. It causes these big clumps to kind of separate into smaller, more manageable clumps. But also, as it's decorating these aggregates with C3b, it allows CR1 to interact with it. And CR1 in this case is in this setting is expressed on red blood cells, and it'll grab these precipitated immune complexes, and it will take them to the liver and the spleen, and it'll allow macrophages to take it through FC receptor and CR3 binding, and those macrophages will eat it and clean it up. So using the opsonins by way of a red blood cell grabbing it and carrying it to these sites, it allows macrophages to clean it up. You can see that here. We have all these complexes. They'll start to allow C3b to bind. C3b can carry them. It can be is a ligand for red blood cells to carry it to locations, and we'll hand it off to be cleared. Now, importantly, you also see complement playing a role in cell clearance. Necrotic cells and some apoptotic cells activate complement, and it will result in the decoration of C3b and C4b. And these are opsonins which allow phagocytic clearance by CR1 or CR3. And what this really does is it helps minimize inappropriate inflammation by getting rid of potentially dangerous cells, right? If you think about a necrotic cell, it's expressing things that should not be expressed. But if the complement system could get activated and could clear it out quickly, you won't have the same widespread activation. So that's good. There are bad scenarios. If you have lots of cell death, this actually creates the setting where there will be lots of complement activation, and it will actually outpace the clearance, and you get big inflammation. Ischemia reperfusion is a great example, because during ischemia, cells are stressed. They don't have oxygen. They don't have glucose, and they start to die or express things that are like, oh, help me, I'm dying. And that's bad, but we have to return blood flow. So it's a paradoxical relationship where ischemia is bad, but it turns out once blood flow returns, it can cause a secondary injury. And that's reperfusion. And so when that blood flows back, now you have inflammatory cells and complement who can see these dead cells and the stressed stuff hurting cells are expressing, and they can promote a wide inflammatory response. Oh yeah, a picture. I don't remember that. Complement can also control viral responses. Many viral surfaces are good activators of complement, especially with C3b deposition through the alternative pathway, because these are, again, some viruses are foreign surfaces that have PAMP patterns. It could also interact with viruses by way of the classical pathway, right? If you have antibody binding to viruses that could, that bound antibody could activate C1 or could allow C1q binding. And in some cases, such as HIV and influenza, there's evidence that MBL can bind some of the host membrane, the envelope modifications that have occurred. Once you get this binding by complement, it's going to cause either enhance phagocytosis, because these complement activation would result in the production of opsonins. And it can also neutralize it, right? If you have stuff all over the outside of a virus, it's less able to attach or enter, or if it's being decorated on the way out of a cell, it's less able to be released. And this gives a good example of the way some complement or some viruses and bacteria, but we'll focus on the viruses, have been able to evade complement activation. Some, such as herpes viruses, they encode for their own complement-like inhibitor or complement inhibitor-like proteins. Herpes viruses express DAF-like and MCP-like and CD59-like proteins that help them avoid complement activation so that they can stay around longer to infect cells. Interestingly, some viruses have evolved to use complement receptors to infect a host. And the best example of this, at least the best example I remember of this, is Epstein-Barr virus, which is associated with Burkitt's lymphoma and stuff. Epstein-Barr virus infects B cells by binding to and invading a B cell through CR2. Right? So that's one way to get around it, is to just get into the cell through the thing that's trying to eliminate you. And so just one brief word about complement deficiencies. We will look at, these will come up or potentially come up. So what I did is table 14.2 provides lots of examples. And we don't have enough time and everything to be able to cover the complete breadth of different diseases that are related to inappropriate complement activation or complement deficiency. So what I did is I just pulled 3 that I think give a good flavor of that. But definitely refer to 14.2 to get that to get that more full experience. So what if you were deficient in C3? If you don't have C3, which means activation goes nowhere. What you often would, or what somebody who's deficient in C3 would have, are severe pyogenic, and pyogenic bacterial infections are those bacteria that are encapsulated, which make them more resistant to clearance by other means. And so because you can't activate C3, you're not going to get lysis, you're not going to get anaphytoxin production, and your phagocytosis is weakened because you're not producing those C3 deposition fragments that become opsonins. And so those bacteria are able to hang around much longer and thrive. Deficiency of C3 is also related to lupus-like symptoms. If you think about it, if you don't have C3 to activate, then you're not decorating those large antigen antibody complexes, so they're starting to accumulate. In addition, you're less able to clear dying cells. There are some cases where you're deficient in some of or one of the proteins that are found in the membrane attack complex, whether that's C5, 6, 7, 8, 9. And one of the good examples of this, because you would have recurrent Neisseria infection. So Neisseria is a species of bacteria that's an intracellular parasite or an intracellular bacterium. And because it's intracellular, you need those infected cells to be destroyed and consumed to clear those pathogens. And this won't happen because you can't lyse those those host cells that are infected. So a little bit opposite to that is deficiency of factor H. Deficiency of C3, you don't have it to activate. Well, what if you are missing factor H, which facilitates regulation of the alternative pathway, or you're missing factor I, right, which is who does the cleaving of our convertases. This is associated with glomerulonephritis and atypical hemolytic uremic syndrome, or AHUS, AHUS. And why this happens is that if you don't have those inhibitors, you actually cleave. There's unregulated C3 activation, which leads to C3 deposition in places where it shouldn't be deposited. Right? There's nothing to slow it down. And what that can cause is impaired clearance and inappropriate host damage, in addition to the fact that you then don't have C3 when you need it, right? And so there's big consequences to that. So just a small flavor of some of those complement deficiencies, and we could spend much more time on that. Okay, I think, yeah, I finished. I expected we would go through that we would not spend, a ton of time on it necessarily. Like, this wouldn't be a long lecture, or as long as it could be. And I think that's important, because what I think this gives you is, at least through the rest of the class period, 40 minutes or so, just to revisit these concepts. Like, go back through, read the follow the pacing of the complement system, because although it can be complex, because you have different pieces that are moving in and out, there is a pattern, right? You find, well, you get comfortable being able to place them in baskets. So if I give you a question and it references C2, even if you have no idea what C2 does, if you remember that it's associated with classical and lectin pathways, but not alternative, that might give you a step towards being able to, to minimize the number of guesses you have to make, or